Right, good day, Junior Techies. Welcome to this video dealing with question 2 of the worksheet, part of the section on DNA and nucleic acids. My name is Mr. P. I'm going to take you through this question. But before I do that, let me just remind you, please, if you have not already done so, get yourself a copy of the complete worksheet, the worksheet memorandum, as well as the theory booklet. Try those questions first, and then have a look at these videos where we take you through some exam technique in analyzing questions and then helping you do the best that you can in preparations for your final exams. All right, so let's have a look here quickly. So the first thing, looking at question 2, is to get some context in the question. So the diagram here represents DNA replication, not to be confused with transcription of DNA. Okay, so it's very, very important to understand the question's context first. And here you can see DNA replication, which means we're looking at DNA making an extra copy of itself. All right, and if we further look here, we know a few things about DNA replication is that this happens inside the nucleus, and then also it is DNA nucleotides that make up that copy of DNA. So we don't want to confuse RNA nucleotides and DNA nucleotides with one another because there are differences, and we need to be very, very specific here because the question is specific. It refers to DNA replication, and therefore everything we refer to in an answer to any type of question needs to be related to DNA. Okay, all right, so there we go. Let's see now. Here, our nucleotides pair in the following ways: C with G, A with T, cytosine, guanine, adenine, and thymine. Those are the pairings that we are talking about when we talk about DNA nucleotides. They are complementary to one another. All right, here we can see that we've got one strand on the right-hand side that is now unzipping to form two strands which are genetically identical. So that's another important thing to remember about DNA replication, is that my two strands that are formed should be genetically identical to one another, exactly the same. All right, so let's look at the questions here. So we look at 2.1, identify the molecules W and U, and if we look at W, already we see molecule W is our DNA nucleotide. We identified that right in the beginning. We know that DNA nucleotides make up the complementary DNA strand. So here we can see that W is exactly that, a DNA nucleotide. Now I know in your memorandum, if you look at your memorandum, it simply refers to nucleotide. Now depending on your examiner, all right, the examiner might feel that simply writing nucleotide is too vague because there are two types. There are RNA and DNA nucleotides. One where we've got our deoxyribonucleic acid. So in other words, there is a deoxyribose sugar versus the RNA where we have a ribose sugar. So here, because this is DNA replication, the most correct answer here is DNA nucleotide when we refer to W. All right, looking at U, we see that U refers to that single strand, and that is our DNA strand. Right. 2.1b again asks us for molecule W and then parts X and Y. So looking at the molecule W, again, we've got DNA nucleotide, but the parts X and Y of this DNA nucleotide, X being our phosphate group and Y being our deoxyribose sugar. So simply writing pentose sugar or sugar is not going to be good enough here. We have to refer to deoxyribose because it is a DNA nucleotide. Bond Z of question 2.1c is our double weak hydrogen bond. All right, it is a weak hydrogen bond with the emphasis on weak. All right. Sometimes the examiner might accept hydrogen bond, but we refer to a weak hydrogen bond holding these together, these nucleotides in their complementary pairs. Here it asks for nitrogenous base V. Okay, if we look at V, we see that V is not really labeled for us in any particular way, but we see that in this particular image, the complementary base is blown up for us as molecule W. And we can see here that the base is thymine. Now, thymine would pair with a complementary adenine. So that's how we would work out the base V, the nitrogenous base V, is we would have to look at the full context of our image and then use that context to answer our questions. All right, brilliant. Let's move on to our next question here. Where in the cell does this process take place? Well, if we look at DNA replication, the DNA is inside the nucleus. If we're creating an extra copy of this DNA, it means that that extra copy would also be present in the nucleus, actually as part of that same now replicated DNA molecule. So we find that the answer to this question is simply the nucleus. All right, so Junior Tukkies, I hope that this video has helped a little bit. Question 2, very straightforward, very to the point, but getting context is so important when you're looking at an image, making sure that you understand exactly what the question is asking you before you answer is also so important, and practicing questions like this will definitely get you that A at the end of the day. So keep on working hard, and again, if you don't have the worksheet and the worksheet memo, get your hands on it. The theory booklet as well, super important. Junior Tukkies, I hope to see you again in future videos. Good luck in your preparations. We'll see you again soon.